Okay. Ready?
Hello everyone and hello from Malaysia. Welcome to our AIMST Biotechnology Students Association webinar series. Thank you very much for your participation today. You have joined our webinar on exploring the boundaries of life using advanced integrative methods. We will begin our program shortly. I would like to kick off today's program by reminding you of some of the housekeeping items as seen on the screen. First of all, please turn off your microphone during the session and only unmute when required to speak. Please turn on your camera so that we can see each other. If you have any questions or comments, please feel free to message us through the chat box. We will try our best to address your concerns. The chat box is also there for you to introduce yourself. Please give us your name and your organization. Last but not least, no phone calls or disruption during the session. Note that this session will be recorded and live streaming is done through YouTube. The recording will be saved on our YouTube channel right after the session. An evaluation survey will appear at the end of our session. Please spend some time to provide your feedback as it will be really meaningful for us to improve our next session. Thank you very much for your attention, and once again, welcome all to our webinar organized by AIMST Biotechnology Students Association. My name is Tan Ying. I'm proud to tell you that I'm Year 2 student of Bachelor Honours in Biotechnology degree program of AIMST University. It is a great honor for me to serve as the MC for this webinar. If anyone has any issue with audio or video, please use the chat box and we will do our best to help you. To start the session, I would like to invite Mr. Hugo, the APSA president, to deliver his welcoming remarks.
Thank you, MC, Ms. Tan. Hello, everyone, and greetings from Malaysia. On behalf of the AIMST Biotechnology Students Association, I would like to welcome our honorable speaker of the day, Dr. Shailesh Datte, who is the Chief Executive Officer of the well-known Laboratory for Research in Complex Systems, United States of America. He is also an Associate Adjunct Professor at the Department of Epidemiology and Biostatistics, UCSF, USA. Senior Professor Manekan Ravichandran, the Dean of Faculty of Applied Sciences from AIMST University. Our AIMST Biotechnology Student Association Advisor, Senior Associate Professor, Dr. Subhash. Professors, lecturers, ladies and gentlemen, thank you all for joining us. I'm glad to see all of you here virtually on this lovely weekend. I'm delighted to know that we have more than 660 registered participants from 22 different countries. We have participants from Afghanistan, Algeria, Australia, Bangladesh, Cyprus, Ethiopia, India, Indonesia, Iraq, Japan, Kyrgyzstan, Libya, Malaysia, Myanmar, Nigeria, Pakistan, Philippines, South Korea, Sri Lanka, United Arab Emirates, United States of America, and Uzbekistan. This is our 6th webinar as a part of our APSA webinar series. As an association of students, APSA always strives to provide a quality and comprehensive social platform to our biotech and bioinformatics students here at AIMST University. We want to help them to learn new things, to grow, and to have fun in the process. We had organized numerous activities, including the annual sport event, APSA Olympics, faculty prom nights, undergraduate orientation, to name a few. In this age of modern technology and the ever-growing field of biotechnology, our intellectual capacity is essential. However, our soft skills are equally important. In fact, I would say that our soft skills are more important than ever before, no matter whether you are a researcher, entrepreneur, patent agent, or writer after your graduation. Therefore, APSA has started this webinar series. All students, APSA members, will get an opportunity to organize a webinar as a part of our webinar series. Our webinar series aims to provide a platform for the students to develop as well as refine their various soft skills, such as leadership, teamwork, communication, interpersonal skills, event organization, and time management skills. These webinar series are really student-centric and helpful for involved students in developing and sharpening their soft skills. At the same time, these webinars are helpful to APSA members and all participants to update themselves with the latest knowledge in their respective areas of webinar topics. I want to thank our honorable speaker again, Dr. Sailesh Datte. In fact, we are honored to host you, sir. Thank you for accepting our invitation and becoming a part of our webinar series. I would also like to thank Professor Manikan Ravichandran for your support. I want to thank the organizing committee as well for doing a marvelous job. The efforts, the time they committed to this webinar are appreciable. Last but not least, my special thanks to our APSA advisor, Senior Associate Professor Dr. Subhash, for taking initiative to start this student-centric webinar series. Furthermore, thanks to Dr. Subhash for training as well as guiding my team members. Ladies and gentlemen, once again, thank you all for joining us. Without further ado, Ms. Tan Yi-Ying, over to you. Please lead the way.
Thank you, Mr. Hugo, for the wonderful welcoming remarks. Next, I would like to call upon the organizing committee chairman, Mr. Lo Bing-Feng, to say a few words and introduce the moderator of today's webinar. Mr. Lo Bing-Feng, the floor is yours.
Thank you, MC Ms. Tan Yi-Ying. Hello, everyone, and hello from Malaysia. I am Lo Bing-Feng, a third-year student of BSc Bioinformatics degree program of AIMST University. It was an honor to serve as chairman of the organizing committee of this webinar. Our today's speaker, Honorable Dr. Sailesh Datte, who is Chief Executive Officer of the Laboratory for Research in Complex System USA. Senior Professor M. Ravichandran, Dean of Faculty of Applied Sciences of AIMST University. Senior Associate Professor Dr. Subhash, who is our AIMST Biotechnology Students Association Advisor, students and staff of AIMST University, and all the participants. On behalf of the AIMST Biotechnology Students Association and the organizing committee of this webinar, I welcome you all and want to thank you for joining us. This is the 6th webinar in our APSA webinar series organized by AIMST Biotech Students Association, and we are proud to host it. I want to thank Dr. Sailesh Datte for accepting our invitation and becoming a part of our webinar series. I also want to thank the Dean of our Faculty of Applied Sciences, Senior Professor M. Ravichandran, for his full support. As chairman of organizing committee, I want to thank every members of organizing committee for their contribution. Last but not least, I want to thank Senior Associate Professor Dr. Subhash. Our committee members are grateful to Dr. Subhash. He has guided my team throughout the process to make this webinar a reality. As chairman, I noticed that Dr. Subhash wants to see each of his students excel. On behalf of APSA and Webinar Organizing Committee, I want to thank Dr. Subhash for his guidance and molding us to play these roles. Ladies and gentlemen, before I pass the mic to the MC, I want to introduce the moderator of this webinar. Today's webinar session will be moderated by Mr. Lo Weng-Hong. Mr. Lo is a Year 2 student in BSc Biotechnology degree program of AIMST University. Thank you, Mr. Lo, for accepting the role of the moderator. Ladies and gentlemen, once again, I welcome you all and thank you for joining us. Let's get ready to listen the thoughts of our today's speaker. Without further ado, Ms. Tan Yi-Ying, over to you.
Thank you, Mr. Lo Bing-Feng. Now, I request the moderator of the webinar, Mr. Lo Weng-Hong, to introduce the speaker and start the session. Mr. Lo Weng-Hong, please.
Thank you, MC Miss Yi-Ying. Hello, everyone. Wherever you are, I hope you're having a delightful day. I'm Lo Weng-Hong, and I'm proud to tell you that I am a Year 2 student of Bachelor of Biotechnology degree program of AIMST University. It is my great honor to serve as moderator for this webinar. I want to thank all the participants for joining us. Ladies and gentlemen, in our daily life, we always deal with various challenges. The challenges that we face may be related to the education or career or making the right diagnosis to boost the health of the elderly parents. To tackle the challenges, we always explore different possibilities and approaches. All of us are living in the modern age of advanced science and technology. How can we make human life better with the help of science and technology? What should we know? How we can use biological system? Furthermore, that can we do to explore the boundaries of life? Today, in this webinar, we will have an excellent opportunity to learn more about exploring the boundaries of life using the advanced integrative methods. We are lucky that our speaker from the United States of America will talk on his vital topic to share his thoughts with us. Ladies and gentlemen, it is my great pleasure to introduce Dr. Shailesh Date. Dr. Shailesh Date is having a very vast experience and lots of credentials. I am going to highlight some important points to introduce him in brief. Dr. Shailesh Date gained his PhD in molecular biology from the University of Texas at Austin in USA. As a postdoc, he did his research work at the University of Pennsylvania in USA. Currently, he is Chief Executive Officer, the CEO of Laboratory for Research in Complex Systems in USA. He is also an Associate Adjunct Professor at the Department of Epidemiology and Biostatistics, University of California, San Francisco in USA. He is a member of many societies and professional bodies, including the American Society of Microbiology. His work entails elucidating first-order principles of biological interaction, both beneficial and harmful, at different scales, from individual molecules to entire cells. This includes examining genes and proteins that are expressed to facilitate the interaction, as well as biochemical and morphological aspects of the process. He is particularly interested in the evolution and development of biological complexity from a system perspective. Over the course of his career, he has examined biological systems of pathogenic and non-pathogenic bacteria, protists, and viruses for their ability to detect and interact with each other, as well as their respective hosts. He has published a number of research papers describing protein interaction networks, their properties, and how they affect function. More recently, his group has undertaken an in-depth study of complexity related to biological systems, focusing on the development of statistical physics models that help integrate evolutionary concepts with physical processes, such as protein interaction. He works closely with a group of collaborators from Caltech, Stanford, Joint Genome Institute, and NASA as part of a team interested in tackling big questions in science. With this brief introduction, I am going to invite speaker Dr. Shailesh Date. Before I do so, just a gentle reminder for all the participants. If you have any questions to the speaker, please post them in the Zoom chat box so that the speaker can answer them during our Q&A session. Ladies and gentlemen, now I would like to invite Dr. Shailesh Date to take control of the floor. Dr. Shailesh Date, the virtual floor is yours.
Yeah. Well, thank you very much. This is really, really interesting. Thank you for inviting me. Subhash and I go back a long way. We were students together, and it's interesting to see him, guide such a good group of students and such an enthusiastic group of students. So, how do you want me to do this? Do you want me to share my screen, or do you want to run my slides, or how do you want to do this?
You can go with your slides, Shailesh.
Okay, perfect. Okay, so I'm going to share my screen. Give me 1 second. Okay, I hope you can see it. Okay. So, one of the things that I asked Subhash was, what is the kind of audience that I'll be speaking to? And he mentioned that there's going to be a mix of people. So what I've decided to do is keep the talk sort of broad so that you can get an idea of the kind of stuff that we're working on. But if any of you have questions even after the talk, feel free to send me an email or let Subhash know, and we can definitely explore things further. So as it was introduced briefly, I want to tell you what my group works on. We are essentially interested in sort of looking at the organizing principles in biological systems, also known as sort of systems biology. We're interested in understanding what components make up biological systems, how they interact with each other, and how these interactions and processes are controlled. We use this information, we use the knowledge that we gain from some of our analyses in various different areas. I have worked traditionally, for most of my career in two areas. One is to look at complexity in biological systems per se. And this includes using various kinds of computational methods, modeling, statistical physics methods, and so on. And we focus it on looking at, life as information and origins of biological complexity and so on. The other area that we focus on is infectious disease. That was my postdoctoral training. I'm quite interested in looking at pediatric health of children, looking at disease transmission in urban areas, in very large urban areas, as we are seeing all over the world today. And you'll be surprised as to how many components are common between some of these large complex systems. So that's our focus. Before I get into the talk, I also want to tell you a bit more about our organization. So the organization is now known as the Laboratory for Research in Complex Systems. And it has gone through a couple of iterations. It was founded by Nathan Wolfe as the Global Viral Forecasting Initiative. And you guys can look him up. He's a famous virologist. And at that time, as the name suggests, the focus of the organization was on epidemiology, infectious disease, fieldwork. There was a lot of assistance given to countries for HIV testing, monitoring of armed forces, and so on. Over a period of time, Nathan realized that maybe there's some value of the kind of work that he was doing in the for-profit sector. So he sort of split the organization in two. One part remained nonprofit, which was renamed Global Vital, and the other part became for-profit, Metabiota. And for some time, both these parts continue to do similar kinds of work. And at this time around, this is about 2015 or so, is when I joined the organization. And at that time, it started moving slightly away from infectious disease and more towards ecology, evolution, and looking at public health. So after a few years of the organization, as I took over the organization first as a CSO and then as CEO, I realized that since we moved away from infectious disease, we might as well expand the scope of what we work on. And now the organization has a very broad portfolio. We deal with problems. We try to solve problems in science, the society, human health, and our portfolio now includes everything from looking at quantum AI to parallel chemistry in silico, to looking at the complexity in biology, as well as startups and spin-outs from the organization. So as you can imagine, we had a whole bunch of projects. Some of these projects in the past, which are no longer relevant, but some of these projects might be known to people who work in infectious disease. And we are very proud of our legacy. Some of these PREDICT and PREDICT2 and CHAMP. A whole lot of work was done in Africa, Southeast Asia, but that's our past. Most of the stuff that we work on right now is ecology, evolution, and as I said, a much broader portfolio of different kinds of projects. What I'm going to talk to you today about is I picked one project out of the portfolio, which is the Boundaries of Life. Boundaries of Life is hands down one of the most interesting projects I've ever worked on. And in fact, it is one of the reasons why I joined Global Vital. And I will also talk about a project that has kind of fallen out of this, which is in the project on information complexity and synthetic biology. We call it the ICS program. And there are other projects that, which are individual separate talks in themselves. Very interesting project called Transitions, where we ask a very interesting question as to why we don't find multicellular bacteria. And there are lots of really interesting and upcoming public health projects. So let's jump into the boundaries of life. So the Boundaries of Life initiative, like I said, again, I want to repeat it because it's one of the most interesting projects ever. And the Boundaries of Life asks a very fundamental question, things that typically are not asked in science. And the question is, the central question in the project is, can we find life? Can we find forms of life, kinds of life that have not yet been discovered? Which is you can give an example, a rationalization through the discovery of viruses, through prions and other organisms, and say that we still have a lot to discover. And we took this question a bit further and said, well, are there forms that we have not yet discovered? They're completely different and might have evolved from very different ways. Evolution might have happened multiple times. All of those interesting aspects of it. And before I sort of go into the details, I want to tell you that Boundaries of Life is a project which is not typically expected to succeed. So it's like playing the lottery. You buy the lottery ticket, but you don't expect to win. What the Boundaries of Life project does is it creates a scaffold. It creates a framework for creating, for thinking farther ahead. We call these, sort of the blue sky projects. For thinking ahead, for developing assays, for developing protocols that don't exist right now, because these protocols and assays are answering a very, very different question. The kind of money that is needed for this, and I keep coming back to money. I'll keep coming back to money rather, because funding is important for science. And some of these projects, especially Boundaries of Life, these are very large projects. Individually, you won't be able to put something like this together. But what we've managed to do is we've managed to create a very special team, and I'll talk about that. From various institutions, as it was introduced earlier. It's a multi-year, multi-institutional, multi-million-dollar project that is aimed at answering some of these fundamental questions in life. We are also interested in knowing, if there are bounds and parameters to life. What are the limitations? We're also interested in asking, has life originated more than once in the universe? Are there parallel streams that we've not yet found? And these questions are very relevant to asking, is there life elsewhere in the universe? And if it does exist, how it might have evolved. Because the project is multi-year, we have divided the project into sort of manageable chunks, different phases. And we have we finished phase 1 in 2019, and we're now in phase 2. And there are various different aims, and I won't go into the details, but just very broadly speaking, assay development remains one. We also had a huge focus on collecting samples from normal and extreme environments to assay them for signs of novel life. And there's a whole bunch of ancillary projects that have fallen out of the Boundaries of Life program. Now, like I said, this is an unconventional sort of research project, and this is not something that you hear described every day. So what I want you to do, especially for the students in the audience, is think as I'm explaining as I'm going through the slides, and you can ask me questions either in the chat or later. And this is especially, one of the things that we wrote into the grant is that we are quite interested in furthering the careers, getting new people involved in sort of blue sky, high profile kinds of research. So rather than just, everyone doing conventional work, okay? Like I said, before, there are lots of people involved. There is a core group of organizations, which includes Global Vital, or LRC, Stanford University, and Caltech. And we also have very strong collaborators from NASA JPL, the Joint Genome Institute of the Department of Energy, San Francisco State University, and a couple of other places. The whole effort is funded by the John Templeton Foundation. They've put in a lot of money into this. It is also funded by the Gordon and Betty Moore Foundation. There are individual projects that are funded by NASA, by NIH and NSF. And my group is also funded by the defense, by the military, the DARPA agency. The funding till date for this project is more than $20 million. And we have many, many members, 20 members or so on a rotating basis. People have graduated out of this project, become postdocs, gotten faculty positions, and so on. So we're very proud of sort of this training aspect that you have. Okay. A quick note about the PIs and advisors. So Nathan Wolf, who is no longer with the organization and no longer with the project, initiated the project. And we have Steve Quake from Stanford, we have Grant Jensen from Caltech, Rob Phillips from Caltech, and yours truly from the LRC. And we have very strong advisors and collaborators, and very thankful to them. Some of them are even co-supervisors for my postdocs. We have Eddie Rubin, who is a former head of Giant Genome Institute. We have Victoria Orphan, who is very well known for deep sea work. We have Ken Williford, who's the deputy project scientist for the Mars 2020 mission in NASA. And we have Tanya Waiki, who is the head of the microbial genomics program at the Giant Genome Institute. Okay, so that's a bit about the logistics. Now the science of boundaries of life. So as I said before, boundaries of life as almost impossible to answer questions. These questions are really difficult. This is not something that that you can answer quickly. How do you essentially go about finding life about which you don't know anything? You don't even know where to start. It is difficult for people to imagine if I say that there might be life that does not have DNA, that is not DNA based. It is difficult to imagine. And in fact, there was a funny story there. When I was, when I came on board, I mentioned to one of the project managers that they probably had a mistake in one of the flyers, and that there is no such thing as life without DNA. And he looked at me and smiled and he said, "That's exactly what we're trying to find." So more food for thought. Essentially, that's what got me interested. We also, and I'll talk more about this, but we also do not want to sort of slide into philosophy. We want to keep doing science. We don't want to tackle questions like what is life? That is out of the scope of this project. We do not want to establish or reestablish any definitions of life. What we want to do is, define very concrete questions and work in areas that are accessible and where we can show progress to the funders and, publish papers and students can move forward. So within this framework, how do you start and where do you go forward? So what we've done is, we've created a conceptual framework that helps us organize and guide the project. And the framework is actually very simple to understand, but it is sort of, one way in which we can tackle this complex question. So imagine that you have, a spectrum of life, and on one side is known life, and on the other side is unknown life. And as you move along the spectrum from known life towards unknown life, life becomes more and more peculiar, more and more esoteric. Initially there is just, life is all similar, things that we know has standard biochemistry, but then, things change, you get extra proteins here, an island there, some kind of, plasmid here, extra plasmids, extra chromosomal elements, what have you, and things move forward. And finally, at some point, it crosses into, it's still DNA based, but it has a lot of novel biochemistry. This is commonly seen, say, for instance, when you're looking at, say, deep sea, life in deep sea, areas, especially where there are active volcanoes under the sea. So, you will find life which has very strange biochemistry. And as you keep moving along the spectrum, it can become even more interesting. And again, like, as I said, I would like, especially the students in the audience, to keep their minds open. And when I make statements like, well, and then we might move into non-DNA based life. So, there's a interesting, sort of definitional play on the definition here. So by non-DNA based life, we mean that life can not be based on DNA, but based on RNA. And we already have life, you know about viruses that are RNA based. So, but what we're asking is, can we find, say, other microorganisms which are non-DNA based, which might be RNA based. So that is another area. And then as we keep moving, we can sort of stumble into areas where life is not carbon-based, silicon-based, who knows what it is, and then completely novel kinds of life. And as you might have noticed, as we are moving along the spectrum, you will find that initially we can work within the parameters of biology. But after a while, biology is not enough. You actually have to start looking at chemistry. And after a while, even chemistry is not enough. You actually have to look at physics. And this is one of the areas in which my group works, essentially the physics of life. We're not the only people, thankfully, who work in this area. There are many groups around the world. I've just listed 3 of the most important ones and 3 of the ones that are most well funded. We have, of course, NASA JPL looking at, through their Mars missions and so on, looking at all kinds of life. In fact, this representation is not exactly accurate. They might be looking at other kinds of life. I just don't know about it. We also have a big foundation called the Simons Foundation that funds origins of life studies. Their focus is particularly on sort of early Earth chemistry, RNA-based life, and so on. And we also have the NELLI program from the Giant Genome Institute, which focuses on new lineages. So they work exclusively in biology, but they're also looking at sort of new lineages on the tree of life that have not yet been discovered. Okay. While we've been doing all of these things, putting the framework, people, all the labs have been looking at assays, developing assays, and there's just too many to list. So what I've done is I've listed examples of what people have done. So for instance, Grant Jensen out of Caltech has pioneered something called high throughput microscopy. High throughput electron microscopy. So typically people spend a day or two, they get electron microscope time, and they get maybe an image or two, or maybe a couple of images of their project. What Grant Jensen can do and his lab can do is they can take more than 5,000 images overnight of any kind of specimen that you give them. So it's an amazingly fast way to scan very large areas, at very small, at very high resolutions. So he has done that. We also have Steve Quake's lab. Steve and his postdocs and grad students have, invented something called mini metagenomics. This is based on their microfluidics platform, and they're able to exquisitely go down into single cells, and they can reconstruct, genomes of individual cells. So again, very interesting work. They have managed to identify thousands and thousands of contacts. They're still in the DNA-based side, but they've managed to identify thousands and thousands of contacts that are completely novel from many, many different environments, including the human and primate blood. We also have, one postdoc of mine, Kevin Friedman, did really fantastic work. He's now a professor at UC Riverside. And he put together 3 different projects actually, and I'm hoping that some of the work will continue. But he started looking at creating mass spec standards, working with folks at, in Florida, to create these standards for something called XNAs. So XNAs are thought to be different non-canonical nucleic acids that might still be found. That might be found in DNA. And the rationale is that while evolution was settling down on A, T, G, and C, there were some of these other things that evolution might have played with, some of these other non-canonical bases that evolution might have played with. So he's put together a project on that, and there's some preliminary data there. He also put together a very interesting project to look at groups of cells, through in microbial communities, which do not appear to have DNA or RNA. If you try to stain them, they do not show DNA or RNA. So we don't know if these are sort of, these high, sort of very advanced, very interesting vesicles, or if there is really some truth there where you don't have DNA or RNA. He also put together another project about, just a simple thing about, lysing cells. And he showed that if you just do different kinds of lysis protocols, you actually get much more information out of, simple DNA studies. And we have tons of papers. This is actually an older slide. We have more than 20 papers now, almost 20 papers now, from all the labs in various different journals, and really exciting work. And I'm not going into the details, like I said before, of any one of these things specifically. Firstly, because it's not my data, some of it belongs to other people, and also there's just not enough time. Each project is a talk in itself. But if you're interested, there are many things here in this list. I would ask you to look at, say, for instance, work done by Grant Jensen out of Caltech, especially looking at cell walls and membrane, especially looking at membranes and bacteria. And what they've shown is that it is likely that the last common ancestor of bacteria was a double membrane entity, and so it might have originated from a spore. And if all bacteria have originated from a spore, it might be possible that spores came from somewhere else and landed on Earth, and life on Earth is connected to other places. So very interesting, keeping with the boundaries of life theme. We've, as I mentioned, we've divided the project into chunks. We're into phase 2 now. Phase 1 is over, but people are still developing assays. And phase 2 is very ambitious. People have taken on lots of interesting projects that have fallen out of the main boundaries of life questions. Grant Jensen and Rob Phillips are looking to develop software to analyze the thousands and thousands of, actually close to 1 million images now of electron micrographs, and to see if we can find something interesting in there. There's also immunotagging, sort of novel membrane biochemistry that is being looked at. We have mirror life, projects around mirror life, XNAs, RNA phages, energetic costs of building forms from the Phillips lab, and also origins of biological complexity from my lab. So that's a bit about the boundaries of life. Like I said, one of the questions I get commonly is, so have you found anything? And no, we haven't found novel life. But in the process, we have developed lots of really cool assays. We've developed workflows. We've developed these integrative workflows where samples get imaged and then they get passed on to for sequencing, and lots of really sort of advanced work on looking at sort of energetics and physics simultaneously. So we have, sort of, for some of the specimens, we have a very, very complete picture. We know where it was collected, how it looks like in all detail, what the sequences are, if it has anything special, what kind of chemistry it has, and so on. And you can read much of the stuff in the papers that are listed. So the next thing that I want to talk about, how are we doing for time? Okay. The next thing that I want to talk about is essentially, the project that is going on in my lab, which is the origins of biological complexity. Typically, at this point, I pause and ask if people have questions, but I think we'll take that towards the end of the talk. So this ICS program in infection, complexity, and synthetic biology, has fallen out of the boundaries of life program. And this is really what most of my group is working on, and most of the people at LRC are also working on. They're funded for this. The goals of this program are essentially to try and develop experimental and theoretical frameworks that explore development of complexity. And one of the hallmarks of life is, ever since life began, I'll show you in the next few slides, ever since life evolved, it has not stayed simple. And that's one of the key questions, one of the most fundamental questions that we still have to answer satisfactorily. And that is that how and why has life become complex? And so we want to explore that paradigm in sort of a very orderly manner, using proper models, and try and see if we can sort of get both computational and experimental projects working together to answer these questions. There are certain assumptions and certain key questions that we are working around. The project can actually be divided into two parts. One is the emergence of complexity itself, and one is, and the other area is what is reflected in the title, which is life as information. How does life accumulate information? So I will not be talking about the second part today. I'll just be talking about the emergence of complexity. We'll ask, I'll tell you how we are trying to tackle that question. And one of the key things that we're also asking is if we can sort of try and introduce synthetic components within this process that can drive, or sort of within our models that can drive the evolutionary process in a certain direction. So this is the famous Miller-Urey experiment, and since it's 1950s, it's been 70 years, and we still are thinking about this. Is how did life go from a few organic molecules that were created probably in a small pond of water through maybe lightning or some other electrical discharge? How did we go from there to something that is as complex as, say, maybe the translational machinery? And this is not even the most complex part, but how did we get from a few organic molecules to something that is so ordered, so stochastically accurate, which is the interactions are random, but they are they're primed, they're temporal, they're spatial, they're very accurately ordered. How did we get there? And that's the question that we are trying to ask. And like I said, it is not, biological complexity is neither well explored nor well understood. And I want to make a distinction here by saying that we're not talking about evolution per se. Evolution, of course, we have explored evolution upside down, but we have, this is the sort of the cousin of evolution, which is biological complexity, and we haven't really explored biological complexity very well. So the key questions, remain is, why is the biological world so complex? Complexity, if you look at energetics, if you look at, some say maybe information theory or even entropy-based, ideas, complexity is not really a prerequisite for survival. You can have simple forms that survive and that are just as fit to survive as some of the more complex forms. So why has complexity come about? How has it come about? And another interesting question is, how do biological systems accumulate and maintain information and this complexity? So, again, a very complex question, pun intended. And anytime, at least the way I go about working is that when I don't, when something is very tough, I try to break it into different chunks and try to analyze each chunk. So that's what I tried to do with biological complexity. And this is just a simple breakdown of its components. And if you think about it, there's complexity at many, many different levels in biology. And this complexity can be of many different types. You can have, say, informational complexity, you can have mechanistic complexity, structural behavioral, there are all kinds of other kinds of things. So if you sit and think about it. But what we are interested in really is this concept that I refer to as bootstrap complexity. And that the example I can give is that imagine that if you're making a wall of bricks, and if you want to make the wall of bricks a bit higher, you expect after, of course, having the groundwork done and so on, after a while, you need certain level of bricks to be there so that you can put more bricks on it and make the wall higher. So this is essentially an example of bootstrap. You need some complexity to be present so that you can build on it. And that is really, this is the phenomena that is really interesting to us. And we have been trying to explore this. Now, the first thing that comes to mind is this is fine, but if this hypothesis is really true, and if you really think about this, can we find examples of this kind of bootstrap? And there are plenty of examples. This is one of the best examples I can talk about is the ATP synthase, structure of the ATP synthase enzyme. ATP synthase has a very unique structure in that it actually has a central shaft that can rotate 360 degrees. So once this kind of architecture was developed, biology was able to take this kind of architecture and then use it for different things. It was able to use it to create the flagellar apparatus, which is which shares the same kinds of architecture and the same kinds of components, and also something very very different and unique, such as the type 3 secretion system. So there is no doubt examples of of bootstrap within biological systems. So what it tells us that when you're thinking about bootstrap, we might as well start thinking about how biology is trying to take one thing, come up with one thing. Maybe the ATP synthase system evolved, synthase evolved for one reason, and then biology realized, or evolution realized, that there was value in it, value in that architecture, and use that architecture for other things. And this is an example, cartoon of a person staring at a pencil, and pencil was made for writing, but the minute you look at it, you start seeing a lot of different uses for a pencil. You can poke people with it, you can clean your ears, all kinds of things. And that is essentially, what our realization is, that maybe bootstrap is working through this adaptation or sort of reuse of certain components that have already been developed. And there are lots and lots of examples of these kinds of of this essential, if you want to call it bootstrap. It's not just ATP, but it's not just the ATP synthase, but the molecule itself. It probably evolved at some point of time and was recognized as a great source of energy. You can use the energy in the phosphate bond to drive a chemical reaction in a certain direction. But the minute the value of this molecule was realized, an entire biochemistry was built around this. So this is, again, sort of an example of how something might have evolved for one specific function, and then biology and evolution took it in a very different direction. And so what we're saying is that maybe once an entity evolves, biology finds new ways, and existing complexity is sort of bootstrapped to create even more complex systems. So that's maybe how it is working. Now, the question for us is, is this something that jives, or is this something that is, that is acceptable to the community? And we're very happy to find that we are not the only people who have thought about this. In fact, this is, this has been published a long time ago by Stephen Jay Gould and Elizabeth Verba, and they even have a name for it. And people have published a lot of papers on it. And this is called exaptation. And as the definition says, something that is a character that was, that evolved based on natural selection for a specific function, which we refer to as adaptation, was then co-opted for some new use. And some of the best examples of this are morphological examples of feathers. So feathers evolved, originally thought to have evolved in heat regulation and display. But soon their value was realized, and they're now used for heat regulation as well as for flight. There are, of course, there are major criticisms of this, because you don't really know where adaptation ends and exaptation begins, and so on, and the boundaries kind of blurred. But this is, we were very glad to see that this is not something that we were thinking, this is not something that that was completely unacceptable. In fact, the community had thought about this, and way before than we did. So now the question for us is, we have this, we are thinking about this. We're thinking about bootstrap. We're thinking about exaptation. Can we actually build a model that can help bootstrap, that can help explain this kind of complexity through exaptation? So that's what my group has been working on for the last 1 year. We call it the biophysical model of adaptive evolution. And I'm sorry, I'm going to jump a whole bunch of steps here. This is actually a big area, and I don't have the time to cover everything in detail. But the key feature of this is, as we were thinking about this, we realized that sort of we were making a distinction between information that is contained in the genome and things that are happening downstream. And we often realize that sort of as we were thinking about the different models, we realized that over and over that genomic information was maybe, like for instance, take the case of ATP synthase. Genomic information was used to encode ATP synthase, but the function downstream was modified or it was co-opted to do something else. So the tool was once made, and then it was used for different things. And there was there's a feedback into the genome, but it is not really needed. What was happening was at the level of proteins, and the proteins themselves were interacting with each other and creating this new use. And so based on this this observation, or this kind of hypothesis, or this structure, we've decided to create this model where we hypothesize that these interactions are the things that change between proteins, and the change in these interactions is actually what gives what allows complexity to build up and new functions to be acquired. And just a simple example, as you can see on the slide, of small round organism. It's a cartoon that has a few interactions. As the interactions increase, the organism also changes morphologically and may develop new features like flagellum and so on. So right now, again, I wish I had more time, but my group and some of the research fellows have been producing some really interesting data. They've also taken this model in a slightly different direction, that of control within biology and so on. But this is sort of an example of of the things that we might find, and we're sort of getting out of the model and into real biology. One of the things that we're expecting to find is examples where, for instance, if you have a single mutation, for instance, here is an example, again, a cartoon of a protein that is found in a membrane. And if there's a mutation, there's some portion of the protein becomes sort of disordered, or maybe, a loop hangs out. And this loop is now free for interaction, and any interactions within the loop don't really cause any conformational change. But the fact that it can now have interactions with other proteins means that it joins an interaction network of another protein, and probably another group of proteins, and has the possibility of providing extra function. And that's how it accumulates. That's how through exaptation, complexity accumulates in a stepwise manner. And there are examples of, say, two-component systems in prokaryotes and so on. Another thing that has fallen out of this model, and again, I apologize for jumping over so many of the details, but another interesting thing that has fallen out of this model has led us to think that we are essentially looking at not just a three-dimensional landscape for evolution, but a hyperdimensional landscape. And as these proteins start interacting with other proteins, develop new interactions, those interactions themselves might become units of selection. And if they are units of selection, then what has happened is the state of the organism has changed, and the organism essentially enters a different dimension. And it kind of, you cannot no longer compare that organism and its state with the state of, its, where it came from, where it originated. So you have a very different set of selective pressures acting on the organism. So that's another thing that we're looking at, and we're trying to see how how we can represent this evolutionarily, and what it means for, say, accumulation of features, and again, sort of the molecular level and biophysical level complexity. So we to wrap it up in some ways, the model explains that, it explains how complexity can develop. It explains extent complexity. It still is within Darwinian principles, selection, fitness, advantages, and so on. Suggests a clear mechanism. And we have also realized that there are multiple implications for the model that we're talking about for synthetic biology. how new biology can evolve. We're going sort of, as as our director likes to say at the LRC, we're going beyond genomics. That's his favorite term. We are no longer tied to the genome simply for information. In fact, we are realizing that biology is actually taking advantage of what it has at many, many different levels. Now, how are we planning to test our model and our predictions? Well, exaptation has remained a concept in theory. There's been a couple of really nice papers that have been published. There's a very, very significant paper published by Andreas Wagner in Nature that examined the evolution of metabolic systems through the lens of exaptation. A lot of people have used artificial life systems to model some of the concepts. All people in the field know that testing exaptation experimentally is very difficult. So what we're trying to do is, we're trying to think through, how we can do both computational and wet lab experiments so that they can inform each other. So one of the things that we're hoping to do is to also use artificial life systems like, Tierra and Avita and all of these, which people have explored. But we also want to do, leaning back on some of my, my work, my previous work in microbiology, we want to see if we can actually stress microbial systems and see if we can introduce new components and then, trace their behavior. So can we model exaptation in existing systems over a period of time? This would be sort of, opposite of what, say, Richard Lenski has done. But we would we would follow a similar path, except under extreme stress, and see if biology if we can actually nudge the evolutionary process in a certain direction. I've also sort of, I've been thinking about this very interesting experiment called, which I call the charm bracelet experiment. So essentially creating a library of random fragments and then exposing, a colony, maybe E. coli or Staph aureus or what have you, to these plasmids or charms within the charm bracelet, and then looking downstream to see, if any of these sort of are taken up by the genome and then used for something very different. One example of this, quite literally, is in some of the gram-negative bacteria, we have some elements that have been taken from other bacteria and that have allowed gram-negatives to survive different kinds of drug pressures. So there is probably value in this, but this would be for the next time when I talk with you guys. Hopefully that I'll be able to share some results with you. And there are some other aspects of it. So without, I think we're absolutely on time. Big thanks to the entire group. I'd encourage you to go and look at the LRC Systems webpage. Lots of really cool work by all the postdocs. Of course, we're very thankful to Nathan for leaving us, with a rich legacy. Our collaborators have been really, really wonderful. They have even taken on co-advising. Tanya Waiki and Ken Williford co-advise my postdocs. It's really wonderful to work with such a great group of people. We also have our colleagues from San Francisco State. I'm also a professor of biology at San Francisco State. We have had junior fellows who were doing their undergraduate work or master's work. And they were, again, brilliant kids. And of course, coming back to the money part, we couldn't have done this without foundations stepping in a really, really big way. So we have the Templeton Foundation, the Moore Foundation. We're very thankful to DARPA, NASA JPL, NSF, all of these foundations. With that, thank you again for inviting me. And like I said, I want to say one thing. So Subhash Subhash and I have known each other for a long time. And one of the things he said is I should probably try and rock you and motivate you. So I don't know if I rocked you, definitely, but hoping that gave you some food for thought. But for the motivation part, I would definitely say, I would definitely give an example of my own career. I started off as a botanist, as a person trained in classical botany and systematics. And I have twisted and turned, I started off with, we got an opportunity to do plant biotechnology when I was doing my master's at the University of Pune. From there, I went to the University of Texas, and within a year, I had switched to a completely different topic, computational biology. No one knew anything about it. No one had known about bioinformatics. No one had even heard about bioinformatics. This was 1999 and 2000, so it was all brand new. And as I was finishing my PhD, I said, I really need to address these infections in humans. And I moved to infectious disease. And from infectious disease, I moved to statistical physics. And so what I'm trying to say here is that it completely is up to you how you want to explore science and what you want to do with your career. And one of the things I'm very grateful for is to be in academic environments most of my life. In academia, like for instance, every person in academia, every supervisor of yours, Subhash, all the faculty, we're very interested in making sure that you find your path, your career. And many people don't know it, but when I was an undergrad, I very cheekily asked one of our professors if I could have my own lab, and they gave me a closet. It was a small space. I got a burner, I got some lab supplies. I used to pour gels. Of course, no one pours gels anymore. I did that. I remember when I was a master's student, I asked one of our professors. Again, this was in botany, and I said, can I take up one of your classes and talk about immunology? And again, she could have, she could have laughed at me and said, that's not what we do. You're studying botany. But she also let me, sort of, discuss for one whole class on immunology. And I think these are the things that I have found a lot of support in academia, and I'm sure that you guys will too. And that's if I can say something about sort of, exploring different areas, then, you should really think about it, jump and try and find new areas. Okay, thanks.
Thank you, Shalistate, for the excellent talk. Now we will have a Q&A session to answer the participants' question. I want to invite the audience to ask the question to clear their doubts.
Okay.
Okay. The first question is, where do the researcher get samples to look for non-DNA or non-RNA life form?
Yeah, so we have, it's a good question because, if there is really different kinds of life that we have not yet found, it can be here, on my computer or on my table, or it can be found in an extreme environment, who knows? So we have made it, we've made it a point to compare both samples from extreme environments and regular environments. For extreme environments, the samples that we've looked at come from lakes that are hypersaline. I mentioned Victoria Orphan. We have some samples from very deep sea volcanic eruptions. We have samples from permafrost collected by a collaborator from the University of Alaska. So these are all the different places where we go to check, to get samples to look for non-DNA, non-RNA life forms. And then we also have, more regular environments, regular ponds, soil, and so on. And so we are looking at both kinds.
Thank you. The second question is it possible to synthesize non-carbon-based protein by using directed evolution on bacteria?
I don't think so. I think carbon is the base. I don't think we can get away from carbon. I'm not sure if I understand the question very well, but right now, no, it is not possible. But you can actually use directed evolution and come up with very esoteric proteins. You can also use one of the projects that was suggested for boundaries of life was to look at something called mirror life. So you have DNL form amino acids, and the question is, why do we find predominance of one form? And there's lots of really interesting theories about why do you find one form, because if you do a chemical reaction, you will find both forms are produced in equal proportions, but sometimes you find a lot of D forms. Sometimes in life you exclusively, I forget which it is, T forms or L forms. So yes, there is interest, but not it still has, everything is still carbon based. We haven't gone to the next level yet.
Okay, thank you, Dr. Shalestate, for answering the participants' questions.
Yep.
It was a really great session. Before I pass the mic back to the MC,
Wong, just hold on. Wong, just hold on.
Yeah.
Can I ask one question?
Yes.
Wong? Lo, can I ask question?
Yeah, sure.
Yeah. So thank you very much, Dr. Shailesh. We enjoyed talk. It was really wonderful and very complex, you know.
I'm sorry if it, I tried to make it, some of the concepts are, but I hope that what I'm thinking is I hope that you guys remember this talk, and then, as you move forward in your career, maybe some of it will come back and maybe you'll be able to ask some interesting questions.
Yes. So still we have a little bit time. So I thought I can grab the opportunity and ask. I have some few questions. You mentioned you are having a collaboration with NASA. So are you working on artificial life forms, or on different planets, or if you shed some light, it will be useful for students. That's what I'm asking.
Yeah. No, this it's a great question. So. I mentioned Ken Williford's name. Ken is the deputy director of the Mars 2020 mission. And one of the things NASA wants to do is, through working with groups like ours, if, for instance, they bring back some samples, they would like to have assays and protocols to analyze those samples. So, and that's what our group is doing. The entire Boundaries of Life initiative is all about. And so that is the connection. So if we do come up with all these assays, then NASA actually has a list of things that they know they can use to analyze, say, Martian soil or something like that. And the fact that we actually haven't found anything, it works both ways. So you have both positive and negative results. So we can say that, if you use, sequencing, or if you use this kind of sequencing, or if you use, electron microscopy in such and such way, you're not likely to find anything new other than probably what is a contaminant of the shuttle. So that's how NASA is interacting with us. Now, NASA does a lot of different things. They have their own programs. They're also very interested in artificial life and all of that. And they're also interested in essentially the bounds and parameters of life. Where can life exist? So we are just one group that are, there's many, many opportunities. For the longest time, NASA had these very large grants that they would award to groups of organizations, groups of institutions, which would then focus on research of interest to NASA.
Yeah, thank you very much, Dr. Shailesh. Another question I have is, because majority of the participants are from biotechnology background.
Mm-hmm.
So what type of advice you want to give to them in this new challenging area, in which area they should go for the career?
There's especially now, when I started, was still, there's academia is very siloed. We do think of botany and zoology and biotechnology. So my advice is, forget about that. There's no such thing. Do whatever you want. If you think that you're interested in bioinformatics, computational biology, do it. If you wake up tomorrow and find that you're interested in oncology, no one is going to hold you back. The world has become open enough that, as long as you have, a good mind and you're willing to work hard and ask good questions, almost any lab in the world will welcome you as a researcher. So don't hold yourself back by thinking, this is what, we have people from all over the world. I mean, especially the Bay Area, California is famous. You will find every single nationality represented here. Because again, if you have the talent and if you're willing to work, then there is no holding you back. So don't worry about individual areas of research. Just jump into things that you would that really interest you.
Yeah, thank you. Thank you. Please go ahead.
I noticed there is a question on COVID-19.
Yeah.
So let me switch to my UCSF mode now, my epidemiology and biostatistics mode. And and including discussions with my wife, who's an infectious disease physician. I do not know about the new mutations. I have heard in passing that South Africa, something in South Africa was, they found something that was, again, I am not sure, but that might escape the vaccine, but we're not sure. Till now, I think everything is on track, and even if you have a variant, the vaccine is most likely to cover it. But we'll see it's going all over the world. Mutations are expected. Something is going to change, and the question is, how fast will it change, and in which direction the change is in. Hopefully, our vaccine is, all the vaccines that have been developed are still going to protect us.
Dr. Shailesh, I got one question from one student. I think by mistake he sent to me privately. So he's saying, is the dark matter of DNA plays any role with the project boundaries of life?
Yeah, actually that's right. So dark matter is a concept. There are two concepts around dark matter in biology. One concept has been looked at extensively by many groups, including people at UC Davis, Jonathan Eisen and his group, and many other people. And that dark matter pertains to genes or proteins, ORFs within DNA that are as yet uncharacterized and that don't seem to have, there's a prediction, but we don't know if there's a protein that's protein that's being made. If the thing is transcribed and translated, but there seems to be a lot of it. And so all this stuff sometimes is considered dark matter. But then there are other definitions of dark matter. One of the definitions that we are using are saying that there is a lot of biology that we have not yet explored, and there is a lot of biological dark matter that we still have to go through and figure out what it is. And then there's some other definitions that people use. So yes, Boundaries of Life is interested in biological dark matter, but a very specific definition of it.
Yeah, thank you, Dr. Shailesh. I have one last question, just for the students' sake, I'm asking, because our Faculty of Applied Sciences wish to send the students to other countries for the internship or summer training. So in your group, you are working with a number of collaborators. So do you receive interns from other countries for summer training for 3 months or something like this?
Yeah, so I don't, as you can, some of the work that I've described, it's not for undergraduates. It is for postdocs and so on. And at that point, we're not really looking for interns. We are actually looking for postdocs. But there are internships. I'm sure there are internships at Stanford. I'm sure there are internships at Caltech. You would have to go about asking individual. The problem with academia, and again, I don't know about industry internships. That's a different matter. But if you want to do, internships in academia, then you have to find someone to sponsor you. Sponsor your, your living expenses and all of that. Oftentimes, faculty, as a practical matter, there is not enough funding in a lab to actually hire an intern and pay them a summer salary or something like that. So, but if there are programs, I had myself looked for internships when I was a graduate student, and it was the same story then. People said, yeah, you're welcome to join our lab, but we just don't have money for you. So if you can find a source of money, if you can find your department to sponsor you. And in fact, I did go to one internship / large course where I was able to find money within the department. So if the department can sponsor you, and I think if you can live with a friend or something, if you can reduce expenses that way, then yes, you can spend a couple of months in a lab. The possibility of learning, though, in sort of high-profile labs is lower, just because they're so advanced that they're the opportunity to pick up like basics is not there. You can pick up advanced stuff, but so you have to choose your lab properly. That's all I want to say.
Yeah, thank you very much, Dr. Shailesh. Lo Weng Hong, back to you. Sorry for interruption because I was thinking I should clarify with you. So
This is
back to
okay,
you.
Dr. Subhash.
Yeah.
Okay. Thank you, Dr. Shalestate, for answering the participants' questions. It was a really great session. Before I pass the mic back to the MC, I just want to take this opportunity to thank the organizing committee for giving me this chance to serve as a moderator. This was my first time in life to serve as moderator. This experience helped me to learn a lot. I enjoyed the role of being a moderator. I must thank Senior Associate Professor Dr. Subhash for his guidance and for being with us through thick and thin. Thank you, Dr. Subhash. Ladies and gentlemen, once again, thank you everyone. MC Miss Ying, over to you.
Thank you very much, Associate Adjunct Professor Dr. Shailesh Tate, for a wonderful and informative talk. And thank you very much, Mr. Lo Wing Hong, for moderating the session. Now, I would like to invite Miss Priscilla to deliver a vote of thanks. Miss Priscilla, please.
Hello, everyone. So I'm Priscilla. I'm proud to tell you that I'm a year 3 student of BSc Honors in Bioinformatics degree program of AIMST University. So the organizing committee has given me this wonderful opportunity. First of all, on behalf of APSA and Webinar Organizing Committee, I want to thank Associate Adjunct Professor Dr. Shailesh Tate for an enlightening and informative talk on exploring the boundaries of life using advanced integrative methods. Dr. Shailesh Tate, we are grateful for the time and effort you took to share your thoughts and experience with us. We really appreciate it. And thank you to Senior Professor Manikam Ravichandran, Dean of Faculty of Applied Sciences, for showing your support to us. Thanks to Mr. Low for moderating the webinar. It was his first time to moderate this webinar, but he did a fantastic job. I want to thank Mr. Hugo, President of APSA, and Ms. Vivian, Vice President of APSA, for their help. Most importantly, I want to thank all the participants from various countries for joining us for this webinar. I believe that this webinar definitely helped you to get some insights and learn something new about exploring the boundaries of life using advanced integrative methods. I would like to take this opportunity to record my thanks to all members of the webinar organizing committee for their hard efforts. Working together as a team for this webinar was really engaging and enjoyable experience. Last but certainly not least, I want to record my special thanks to APSA advisor, Senior Associate Professor Dr. Subhash, for his valuable efforts for creating this wonderful opportunity for us. As students, we are learning a lot through this webinar organizing exercise. Dr. Subhash, we highly appreciate your efforts. Ladies and gentlemen, once again, thank you. And Ms. Tani Ying, over to you.
Thank you, Ms. Priscilla. It was a great session today. Before ending this session, we would like to hear feedback from all participants by scanning the codes on the screen right now. We promise that the evaluation survey will not take you more than 2 minutes. Your feedback will be useful for us to improve our upcoming webinars. You can either scan the QR code or go to the link shown below. The link is posted in the chat box as well. After the completion of the survey, you will receive an instant participation certificate in a short while. And I will keep this slide running for 2 more minutes for you to scan the QR code. We are looking forward to having you all joining us again in the next session. Thank you again, everyone, and we wish you good health and safety.
Okay. Let me know if you want me to stay on or log off or.
Yeah, just hold on, Dr. Shailesh. We'll capture a virtual photograph at the end, you know.
Okay.
Yeah.
Absolutely.
I hope everyone has the chance to scan the QR code, and I'm going to end the meeting. See you soon in our next webinar. Stay safe and have a wonderful day. Bye for now.
Bye.
Bye. Thank you.
Shailesh, just hold on. I think organizing committee, you can take the virtual photographs. We are not physically in one hall, you know. So feel free, you can capture the screenshots. You know?
Okay.
Yeah. So everybody can smile. We can capture photo. You know? New normal, you know, Dr. Shailesh.
Yeah.
Smile, please. Others can open the camera. Please feel free to open the camera. So we will be in photograph. Smile, please. Okay, great. So I leave it to chairman. So chairman, anything else? Because speaker has to leave, chairman. APSA president, you go.
I think that's all.
Yeah.
Thank you, Dr. Shailesh.
That's okay. Okay, guys, have a great day. You know, thank you for coming. You know, thank you for spending your weekend.